Hey, Mr. P here. In this video, we're going to talk about prokaryotic cell wall anatomy. So in past videos, we've talked about some basic similarities and differences between gram-negative and gram-positive bacteria. Well, in this video, because we haven't done so yet, we're going to kind of dive in and talk about the actual composition of the cell wall, kind of in general, and then also some similarities and differences of the anatomical features of the gram-positive and gram-negative cell wall. So this video is all about prokaryotic cell wall, not getting into any kind of eukaryotic cell wall discussion, but all about prokaryotic cell wall. Let's go. So when we talk about prokaryotic cell wall, just like we kind of hinted at last video, is a boundary on the outside of the cell membrane that is going to possess some things that are polysaccharide in nature, maybe some protein in nature, but it's going to ultimately be protection, structural, support, shape, those kinds of things. And so we're talking about this particular structure right here in this particular bacterium. Okay, it's kind of this brown color. It's on the outside of the cell membrane, in this case marked plasma membrane, but the cell wall, kind of at its base function, is a boundary that both protects the cell and prevents osmotic lysis. Now, osmotic lysis is rupture by osmosis. Okay, a lot of times we use in our eukaryotic multicellular bodies a form of osmotic lysis in order to help prevent pathogenicity or help prevent us from contracting disease, especially infection from these microbes. But this cell wall in a lot of cases are going to keep the microbes kind of safe and healthy even in times of immune response. These cell walls often contribute to bacterial pathogenicity. Basically, that means they contribute directly to the bacteria being pathogenic. They are made of peptidoglycan. So peptidoglycan is a structural polysaccharide. Bacteria specifically are going to use peptidoglycan as the base component of their cell membrane. And this peptidoglycan, which we're going to talk about in a minute, is made of repeating disaccharide molecules NAG and NAM. And so you can see those down here. NAG stands for acetylglucosamine, and NAM stands for acetylmuramic acid. You will have repeated NAG, NAM, NAG, NAM, NAG, NAM if we continued or extrapolated out this image. But you have these repeated bands of these disaccharides that come together and are bound by this covalent bond to form polymers, in this case a polysaccharide. Okay, so just right off the bat, cell walls in prokaryotes case are going to be on the outside of their cell membrane. They're going to help protect the cell by preventing osmotic lysis. They're going to contribute in large part to the pathogenicity or the disease causing or the ability to cause disease within a microbe. And they're made of the polysaccharide peptidoglycan, which is made of repeated disaccharide bands, NAG and NAM. Okay? So, if we kind of look at a different view, we will see that these bands, which are colored kind of this brown and salmon color, there are these repeated bands of NAG, NAM, NAG, NAM, NAG, and you have these actual kind of bigger components, which are called carbohydrate backbones, that makes the polymers, okay, which is just a molecule that's made up of monomers. In this case, it's made of dimers. And there are these other polypeptide or protein components which are side chains and cross bridges. Okay, so these peptide bonds, okay, or the peptide bonds are going to hold the individual peptides together, but these tetrapeptides, tetra means four. Okay, so it is a protein that is composed of four peptides, four actual individual amino acids are attached to NAMs in the backbone. And so you'll see that they would be basically attached to all of these salmon-colored acetylmuramic acid dimers. Okay, so these tetrapeptides are always going to be attached to this salmon-colored NAM dimer. Okay? You have these cross bridges, which are blue in this case, and so adjacent rows are linked by these polypeptides, which are protein portion. They are the peptide cross bridges. Okay, they're blue, and so these are the cross bridges that are actually going to link the individual rows. This is a row, this is a row, this is a row. So you have the tetrapeptides, which are kind of linking our rows vertically, and then you have the cross bridges that are linking our rows horizontally. Okay, parallel tetrapeptide side chains may be directly bonded to each other or linked by a peptide cross bridge. Again, all of these proteins, which are these kind of beaded structures, are holding our polysaccharide rows together. It's basically creating a lattice, which is going to give the structural components to our polypeptide, or our polysaccharide in this case, okay, our peptidoglycan. Alternating NAM and NAG molecules are linked in rows of 10 to 65 sugars to form a carbohydrate backbone. This is the glycan portion. So we have all of these protein components. We have these individual dimers that are linked by covalent bonds that are forming our polysaccharides. And so this prokaryotic cell wall, just right off the bat, is really composed of sugar and protein. Okay? So penicillin, probably or hopefully a term you've heard before. Penicillin is one of the first, if not the first, antibiotic molecules that was kind of discovered and used on a commercial scale to help protect us against infection. And penicillin, while I bring this up, penicillin directly interferes with the final linking of the peptidoglycan rows by peptide cross bridges. As a result, the cell walls greatly weaken and the cell undergoes lysis. So basically what this means is penicillin, when we take penicillin, the compounds in penicillin are going to interfere with the final linking of all of these protein cross bridges, which is going to, as you can imagine, cause these individual rows to kind of disassociate or fall apart or get weakened. And if the cell wall gets weakened, then all of a sudden the bacteria isn't protected as well. And if that happens, then the osmotic lysis or the bacterial lysis, the cell wall or cell membrane lysis can take place. That's really important for us when we talk about kind of, staving off infection. So the cell wall in both cases have relatively the same structure, but you will see, hopefully right off the bat, also that there is a substantial difference between the thickness of a prokaryotic gram-positive cell wall and a gram-negative cell wall. Okay, gram-positive cell walls are going to be incredibly thick, and a lot of those protein cross bridges, a lot of those repeated polysaccharide rows. Okay, and the gram-positive cell wall is actually on the far outside or the far exterior. Okay, the absolute exterior of this particular cell membrane. Cell membrane down here. Cell wall is on the exterior of that cell membrane. Now, how does gram-negative cell wall compare? Gram-negative cell walls are actually very thin, and they are sandwiched between two layers of phospholipids, or two phospholipid bilayers. So a gram-negative cell has an exterior cell membrane, they have an interior cell membrane, and then they actually have a really thin cell wall sandwiched between those two membranes. Okay, so if we focus in on gram-positive cell wall, you will see all of the structures we just talked about. Okay, the cell wall is made of thick peptidoglycan, which is composed of teichoic acids, which you can see here. Okay, they're these little kind of squirrely curlicues, and those are also to help hold and give structural support to the rows of polysaccharides. You have all of these teichoic acids are linked to our repeated rows, which is peptidoglycan. These teichoic acids are going to carry a negative charge, and they regulate movement of cations, which are positive ions, specifically into the cell and out of the cell. You have lipoteichoic acids, which are going to link the wall to plasma membrane. Again, you can see that this lipoteichoic acid is going to actually be embedded in the membrane. It's going to link the membrane to the cell wall. It's what gives one structural support to the cell wall, but also adds rigidity from the cell wall into the membrane. Polysaccharides and these teichoic acids are going to provide antigenic specificity. Why is that important? Okay, the antigens are specific to specific cells. Certain bacteria and certain cells are going to actually have antigens on the surface of their cell that is protruding out of the cell membrane, and that's ultimately what our immune response and our antibodies are going to identify as being foreign. That obviously gives our immunity an understanding of what is actually causing infection and kind of tells us and tells our immunity how to handle them. Okay, again, gram-positive bacteria are going to have very, very thick exterior cell walls. If we look at gram-negative cell walls, you will see that they have roughly the same types of structures within their cell wall, but you have an external cell membrane and an internal cell membrane, and you have a very thin peptidoglycan cell wall. So this lipid A is an endotoxin embedded in the top layer. Okay, lipid A is right here. It's these endotoxins that also provide some of this antibody or antigen specificity. O polysaccharide functions as an antigen. Again, I just said that. Okay, lipid A is an endotoxin. O polysaccharide is going to be protruding. That is going to serve as their antigen. That's what our antibodies are actually going to identify as being foreign, this being an antibody of our immune response. Okay. You have porins that form channels through the membrane. So these pores are going to actually cause openings between both membranes and the cell wall that allows substances to get into and out of the cell. Outer membrane made of polysaccharides, lipoproteins, and phospholipids. Again, it's going to be roughly the same composition as our internal membrane, but there is a little bit of extra stuff, only because it is totally on the exterior. This one is a little more protected than this one. And the periplasm is the space between the two membranes where the actual cell wall is embedded. Okay, gram-negative cell wall protects the bacterium from phagocytes and complement and antibiotics. Because the gram-negative cell wall is embedded between two phospholipid membranes, it's a little more protected, obviously, and so a lot of the compounds that we use in our immune response and through antibiotics don't get to the level of the cell membrane or get to the level of the cell wall, and so they can't really cause the same level of lysis that a gram-positive bacteria is subjected to. Okay, thin peptidoglycan outer membrane, periplasm space, and teichoic acids are absent in this particular cell wall. Now, why would those teichoic acids be absent? It's because it is a very thin cell wall. We don't need the teichoic acids to link or give structural support vertically because we only have really one row of those polysaccharides. Okay, so when you look at those membranes, you can see the membrane on the outside, membrane on the interior, and you have that really thin brown cell membrane or cell wall in a gram-negative bacteria. Now, gram stain technique is a technique we've used in lab already, but if we kind of talk about why the gram stain technique works and how it works, this is a little kind of quick demonstration on what we're actually doing when we're employing the gram stain technique. Remember, the gram stain technique is a differential stain technique, so we are trying to differentiate between two different types of cells. Okay, we're not trying to identify specific genera or specific species, but we're trying to differentiate gram-positive from gram-negative bacteria based on just the composition of their cell wall. Gram stain technique first employs our primary stain called crystal violet. Remember, crystal violet is a purple stain. It is a simple stain. It's going to stain any and everything. In this case, we employed the crystal violet over the top of our gram-positive bacteria and our gram-negative bacteria. Okay, gram-negative sitting over here, gram-positive sitting over here. Both of the stains, or both of the samples at the end of our crystal violet step will appear purple because we've only used a purple stain. Our second step is we're going to apply the mordant, and that is going to cause the stain to set in to our gram-positive bacteria. It will slightly set in on our gram-negative bacteria, but because of this external cell membrane, it's really not going to get to the level of the cell wall, and so it's not going to totally set the stain in a gram-negative situation. After the iodine, we're going to wash or decolorize with alcohol. And so what this is doing is it's actually dissolving that outer membrane. And because we dissolve the outer membrane, it is also going to pull this purple stain out, and it's going to kind of dissolve the cell wall because the cell wall is not totally protected anymore. It's exposed because that outer membrane is gone. Now all of a sudden the purple is removed, we've decolorized. We do not, just as a reminder, do not decolorize the gram-positive bacteria, and that's because the mordant caused this stain to really set in and become permanent in a gram-positive situation. But at the end of our decolorizing technique, or our decolorizing step, the addition of the gram-negative, okay, or the addition of the alcohol, crystal violet will still be present in the gram-positive bacteria. It will not be positive or not be present in our gram-negative bacteria, and so our gram-negative bacteria will be colorless, and our gram-positive bacteria will be purple. Now we add our counter stain, which is safranin, and because this outer membrane is gone, okay, we apply the safranin to both cells. Notice that the red kind of overlaid this purple, but because the purple was already present, it really didn't cause it to go red. It just kind of causes it to go maybe slightly red or purple, but the purple is definitely still present. Because we remove this outer membrane, now the cell wall of the gram-negative bacteria is present, and so the safranin sets into that particular cell. And now we have total differentiation between gram-positive bacteria and gram-negative bacteria. Gram-negative bacteria being red, gram-positive bacteria being purple. And when you look at these cells under a microscope, you should be able to see right by, side by side, red bacteria and purple bacteria. Purple being gram-positive and red being gram-negative. So you can totally differentiate, or should be able to differentiate at this step. Gram-positive, again, are going to be purple or appear purple. Gram-negative are going to appear red. Okay? Now, how does lysozyme specifically damage the cell wall, and what is lysozyme? Lysozyme is an enzyme component, okay? It's an enzyme, and it is designed to break apart the alternating bands of NAG and NAM. Okay, we've already talked about what NAG and NAM are. Lysozyme is an enzyme that we use in our bodies in certain spots to help prevent infestation or infection of bacteria. So lysozyme is a compound, it's an enzyme, it's going to be added to the bacteria, and it's going to start breaking these bonds between the repeated bands. And if you break the bonds between the bands of these NAG and NAM molecules, then all of a sudden the polysaccharide is going to be chopped up, so to speak, into these individual dimers. And as you can see, if it's not linked together anymore, there's no structural support to the cell wall, and that cell is subjected to osmotic lysis, or can just be decomposed or digested by our immune response. So where is lysosome contained? Lysozyme is contained in sweat, it's contained in tears, it's contained in all of our mucus linings, and it is contained within saliva. Why is it contained in these areas? These are the direct portals to the interior of our body. Okay? Perspiration helps to coat our entire skin, which is our normal kind of physical boundary between our environment and our interior body. Tears obviously are going to protect our eyes, which are an opening that bacteria could easily get into. Mucous membranes, all of the openings or portals into the body. And then saliva, while it helps to chemically and mechanically digest food and molecules, saliva actually is a portal to our interior organs. And so if we can consume bacteria, or if there's bacteria on things that we put in our mouth, or if just by happenstance, bacteria gets into our mouth, we have to have certain lysozyme enzymes in our saliva that help prevent bacteria from, really taking over and, causing infection. Gram-positive bacteria are extremely vulnerable to lysozymes since they have a very thick peptidoglycan and no membrane to protect the wall. Remember, their cell wall, while it is very thick, and while it might seem like it's very supported and very structurally sound, and while it might seem like a more impenetrable structure, it is very vulnerable to lysozyme only because it is on the exterior, and lysozyme is very good about breaking down these polysaccharides, which really is what the gram-positive cell wall is all about. Okay? Gram-negative cell walls are not as subjected to the lysozyme, and that's because the lysozyme just physically can't get to that particular cell wall, right? There's a membrane on the outside of it, and so it doesn't have direct access to the cell wall in a gram-negative bacteria, and so it doesn't work quite as well. But if you have any questions, bring them to class. See ya.